Mahalo and aloha, everyone. Morning, it's a real pleasure to be here. I'd like to thank the organizers of this session for inviting me. So I first have to declare that I have nothing to declare. And the outline of my talk will be to say why do we study the brain in animal models using preclinical MRI? What are the advantages? I'm going to show the marmoset as a favorite model in my lab for functional brain activation, in particular because we can image them the same way that human brain is studied in conscious awake conditions. And then I'm going to give 3 examples in this talk, a short talk about how we can combine MRI with other techniques for learning more about the brain. So the first one is in combined fMRI and electrophysiology in a marmoset model of visual stimulation. The second one is combined fMRI and optical imaging in somatosensory stimulation. They both use the marmoset. And then in the third example, we're going to show some work of my collaborator Xin Yu at the Max Planck Institute in Tübingen on doing fMRI and optogenetics in a rat model of somatosensory stimulation. So there are lots of advantages of working with animal models, and I would like to start, because of this meeting here, with the technical advantages, which is that we can put the animals in bigger and better toys. On the technical side, magnetic field strengths go up to 21 Tesla in the vertical direction or 17.6 Tesla in the horizontal direction. And we all know that more B0 means more signal that you can use for better spatial resolution or temporal resolution. Gradient performance is also much better because of the small diameter of these magnets and therefore the small diameter of the gradient, so easier to achieve these high gradient strengths that are very important, for example, for DTI type of acquisitions. And all of this then translates into us being able to get some money in the bank for using it to work with this small field of views because of the small brain sizes of the animal models. So that means that we have improved spatial resolution. Now we are routinely working at about 100 microns or better in plane resolutions, which allows us to put lots of pixels and visualize the cortex of the animal and very fine white matter tracts, even in a mouse that has almost no white matter. So the quality of the MRI images are almost histological and allow a direct comparison between MRI and gold standard histology. But also, the main purpose of working with animal models is that we can explore the brain in ways that we can still not do in humans. And that means that we can marry the many advantages of the non-invasive MRI technology with more invasive technologies like electrophysiology or optical imaging that requires a direct visual access to the brain or optically clear access to the brain. And the later array of technologies like optogenetics or chemogenetics that allows you to directly interfere with genes in a way as to express fluorescent or molecular markers that will allow you to detect events before and after the expression of these markers in the same brain. Plus, in addition, we can still perform traditional pharmacological manipulations that allows you to modulate specific enzymatic pathways in ways that are quite specific. And there are several examples on the literature and even on this meeting that you're going to see this marriage of fMRI with these other techniques. And because this is a short talk, like I said, I got 3 examples. So first, I'd like to introduce to the ones of you who don't know what marmosets are. Marmosets are new word, non-human primate, that one of the main advantages for us who like to image the brain is the fact that the brain of the marmoset is smooth. And because if it is smooth, it makes the localization of brain function in the cortex, either with fMRI or optical imaging or even electrophysiology, quite trivial. You don't have to deal with the convoluted folding of higher primates and humans. But in spite of having this smooth cortex, the marmosets retain the typical anatomical and functional organization of the primate brain. And this is very important because then we can do homologous inferences from data that we acquire in the marmosets to data that have been traditionally acquired in the higher order primates, such as the macaques, and then hopefully learn something more about the human brain, which is the end goal. Of course, we can image marmosets, in different types of setups. The traditional one is because animal models are usually non-voluntary volunteers, then you need to resort to anesthesia as a way to alleviate the stress of the animal and also to achieve the necessary restraints so that you can obtain high quality data. And so working with anesthetized animals, isoflurane is the typical anesthesia of choice because it's easy to administer. The animals tolerate it really well and, more importantly, recover really well at the end of the scan. But isoflurane has a lot of problems in interfering with neuroactivity and also with the vasculature in different ways, in ways that are hard for you to control. And they, in many instances, preclude its use for functional studies. There are other ways that you can use anesthesia for functional studies. For example, in non-human primates, this combination of propofol and opioids such as fentanyl work really well in preserving the vascular reactivity and also in preserving neuroactivity. But, in the end, you really are studying the brain of an anesthetized animal from the functional point of view in a state that is not the state that human data is acquired. So because of this, our lab has put a lot of effort into eliminating anesthesia altogether and working with conscious awake animals so that we can do both anatomical or functional studies in the marmoset. So this is a picture of the marmoset in an awake preparation ready to go into the magnet. And regardless of what's the setup we have there, we still put a lot of effort into providing extensive physiological monitoring of them, controlling their temperature, heart rate, pulse oximetry, and tidal CO2 so that we know that they're happy from a physiological point of view inside the magnet. For doing the imaging in the awake conditions, we use a specialized cradle that has a body cover so that the animals dress this sleeveless jacket, and we zip tie that to the body cover so that the animal cannot escape through the back or the front. And then for immobilizing the head, we rely on 3D printing technology to make these custom designed helmets where the helmets are individualized. Each animal has its own helmet so that the conformation of the anatomy of the helmet to the anatomy of the animal is perfect. And when we do this, then the animals almost don't feel that there's something holding their head, so comfortable that it is. This is an example of a marmoset awake, ready to go into the magnet with electrodes attached to its front paws so that we can do somatosensory stimulation. And then once the animal is in the magnet, you have the receiver coils. This is an old picture. There, the animal is just fine, and they do exactly what human subjects do when they go in the magnet. They take their chance to take a nap. So much for awake scanning. But this setup has improved quite a lot so that we can now present stimuli inside or outside the magnet. So in the case of visual stimulation, we have an LCD screen outside the magnet. We have an eye tracking camera that looks whether the animal is looking at the screen or not. The coils themselves became quite specialized. We are now able to put the receiver coils inside the inner surface of the helmets for maximum sensitivity. And we can deliver rewards to the animals to incentivize them to participate in the task and obtain better performance this way. So this is an example of these in-helmet embedded RF coil arrays that we designed. These are low profiles, and the sensitivity is really good. These are anatomical images combined with fMRI, acquired at 250 microns in plane resolution of the somatosensory cortex of the marmoset because of the added sensitivity. So the first example that I'm going to give here is using fMRI and ElectroCorticoGraphy ECOG arrays doing visual stimulation. One of the things that wasn't known about marmosets is whether their visual pathway would have specialized areas for detecting faces versus other objects. So we did a block design where the stimuli composed of images that were classified into different structures such as faces or body parts or objects, and we had also some controls. And we did a block design paradigm where each block would have stimuli of one of the different categories. And together with the fMRI, we would have ECOG arrays implanted in their temporal lobe to verify for the presence of these areas that have preference for faces. So here's a little bit more on the block design paradigm. Each block was 16 seconds of the stimuli of the same category. And if the animals were looking at the screen for 1.5 seconds, they would receive a drop of liquid reward as a positive reinforcement, no negative reinforcement. That's just to incentivize the animals to be looking at the screen. And here's an example of the animal. This is the tube that brings in the reward and the eye tracking camera. There's no stimulus, so the animal is looking anywhere. You see how the gaze is varying all over. And then once the spot there comes, you see how fixed the gaze of the animal is now. And the stimuli come and the animal starts receiving the reward. So it's like really a positive kind of task for the animals. They enjoy participating in this kind of task. You see how the animal is maintaining its gaze on the screen really well. And then once the stimulus disappears, you immediately notice that the animal is saccading all around. Again, the reward stops, and then the animal goes back to its resting condition. And with the data that we acquire with this kind of experiments is really outstanding. We can see activation of primary, secondary, tertiary, and higher order visual cortices with lots of properties that I don't have the time to get into here, but data that really has very strong homology to data that is being acquired in macaques and the human brain. And one of the particular data that we obtained for the first time in marmosets is revealing this face-selective patches that have that they are discrete in nature, like in macaques and in humans, and that they have higher activation when the stimulus is faces versus any other stimulus. And this data is now published both in the journal Neuroscience and also in Neuroimage. But on the electrophysiology side, using the same kind of paradigm, we're able to get these event-related potentials that also vary according to the stimulus categories. And then we get this analytical amplitude of it. And then depending on the location of the electrode, it will have preference for faces versus body parts or objects. And this is something that follows the duration of the stimulus. And we can then get this analytical. So this is a face preferring patches. And when we make a map of all the electrodes that are around the temporal lobe, we see that there are areas that like faces more, like electrode number 3 or number 4, versus areas that don't like faces, like electrode number 8 here, for example. And if you make a map of this face preferences in green here versus what we obtained with fMRI, you see that the correspondence is really remarkable. So this is an example where you really see the electrophysiology conforming data obtained with the fMRI. A second example is to go now into optical imaging and to try to directly visualize the neurons and the blood vessels in action, so that we can better understand neurovascular coupling. So in the mouse cortex, you're able to label the neurons with this calcium indicator, GCAMP, and you see that every time the neurons fire, you have an increase in fluorescence that you can easily detect with the microscope. So you can do that in a marmoset as well by doing a cranial window to allow direct visualization of the cortex. And one of the features is that we have a very long stability of their preparations, so we can get more than 6 months worth of recovering before we lose optical clarity because of regrowth of dura in the cranial window. But so if you inject then the cortex with a virus that expresses the GCAMP molecule, here's the area of coverage, you see that post-mortem that the virus works and that the neurons express GCAMP molecules in there. And then when you do the optical microscopy, you see both blood vessels with an intravascular fluorescent tracer in red, but you also see the neurons in flashing in green there, indicating their activity. And you also see some of the problems that we have with the motion of the animal during the acquisition. Even though, you know, we have a head post there, you still have a lot of problems dealing with this motion so that you can properly detect their activity in an individual cell. But then if you do an ROI at a single cell, you know, when you stimulate the animal, in this case was somatosensory stimulation over the somatosensory cortex, you see an increase in delta F over F with the stimulus duration that indicates the activity of the neuron. And then in a blood vessel that is right next to it, you can look whenever the stimulus is on that this capillary, it increases in diameter, and it will even vary its diameters in between stimulations, but you have a time-locked increase in diameter. So relating the neuroactivity in the neurons with the neuroactivity in the blood vessels is something you can do well. And then finally, in the third example, with optogenetics, we're going to hear from the leaders of the field here, in particular Dr. Lee, who started this in 2010. She's going to talk here, but I'd like to bring data that is being acquired by Shane Yu at the Max Planck Institute in Tübingen in rats, where he uses an MRI technique, a line scan base, so that he has excellent in-plane resolution of about 50 microns with a 1 millimeter slice thickness, and he can visualize individual arterioles and venules with a gradient echo type of sequence, so that he can look at the BOLD effects or the blood volume effects in this map. So for example, in a BOLD effect with a pixel focused on cortical venule, a principal cortical venule, you see that when the stimulus starts, you have the activation of the capillary bed around it right here, and then you have the principal cortical venule draining the blood flow away. And you know, we now have single vessel resolution in the fMRI. You can also do that with iron oxide intravascular tracer for blood volume maps, and in this case, the voxel now is on an arteriole, so I'm letting it loop again so that the stimulation starts, and once it starts, we're going to see again activation of the capillary bed and then the arteriole here, indicating that, you know, you have the capillaries, which are the blood vessels closest to the neurons, initiating the hemodynamic response in that functional area. So this can be done in combination with optogenetics. So Shin put a fiber optic there, and he uses an AAV virus that expresses channel rhodopsin, and he did all the control for making sure that with electrophysiology, whenever he stimulates the fiber optic, the LFPs have the right shape and amplitude, and he did optimization of all this so that when he pulses light through the fiber optic, he gets a very strong increase in fluorescence in that area in there. And then with the fMRI, this is the BOLD. Whenever he stimulates the fiber optic, then he gets the positive increase in the capillary bed and also in the draining venules. And with CBV, the increase in the fiber optics, in the capillary bed around the optical fiber, and also increase in the penetrating arterioles. So the data, again, of optogenetics marry really well what is being discovered with fMRI, with the advantage of the specificity for the cells because they are the ones that express the channel rhodopsin here. So in conclusion, I hope I've given you a taste for why animal models are important. The data acquisition can be performed in state-of-the-art MRI scanners, and that translates into very high SNR, high spatial resolution, high temporal resolution. So it's the best of all worlds. And they can be married to these comprehensive multimodal investigations. There are lots of challenges, though. Anesthesia, in my opinion, is a major confound for all the fMRI studies done in animal models, and it would be really great if we found effective ways to not use it. And that would require, of course, training of the animals to perform specialized tasks, that brings in the complexity of training the animal, but also the complexity of making sure that you don't have motion associated with these tasks as well. We strongly believe in the marmoset as a very important experimental animal model for both basic neuroscience and translational research. There is also a lot of work in marmosets, if you want to look in this meeting related to using fMRI to understand the marmoset model of multiple sclerosis and of stroke. I would encourage you to go look for it. One of the advantages of the marmosets is that, like rodents, they have a smooth cortex, which facilitates the localization of functional areas and facilitates the study of the neuronal circuits with optical imaging techniques. And finally, one of the reasons why rodents are used so much with respect to non-human primates is the fact that it's easy to obtain a transgenic rodent. So luckily for all of us, these gene editing techniques are now amenable to non-human primates. There are non-human primate animal models now available out there, which I believe will strongly boost their value in neuroscience research, allowing both the chronic and acute monitoring of in vivo neuroactivity. I would like to finish by acknowledging the folks in my lab who actually do all the work. I get to have the fun of being here in Hawaii and communicating this to you, but they are back home doing all this hard work, and I thank you for your attention.